Hello and welcome to the Meat Institute's webinar, Rethinking Food Safety, Science Drives Strategies for Animal Protein Industries. My name is Kate Rose McCullough. I'm the Senior Director of Science and Public Health and President and Chief Scientist for the Meat Foundation. I'll be introducing today's speakers and moderating the discussion for this webinar. We're looking forward to really having a great interactive discussion with all of you on a topic that I know is tried and true on top of everybody's minds. Before we get started, I want to start with a very important antitrust reminder. Just this brief reminder that the Meat Institute and all our participants are expected to comply with all the applicable antitrust laws. This program is for education and general industry information only. Please keep all questions and comments focused on today's topic and avoid any discussion of company-specific prices, costs, business plans, or other non-public competitive information. Our Q&A is moderated, and if a question heads in an inappropriate direction, I will redirect it. Thank you for helping keep our programming compliant. Now, before we begin, I'd like to give a huge thank you to bioMérieux for sponsoring today's session, Rethinking Food Safety, Science-Driven Strategies for the Animal Protein Industry. bioMérieux's continued commitment to advancing science-based solutions and supporting meaningful dialogue across the food safety community helps make conversations like today's possible, and we're super grateful for their partnership, their scientific leadership in this critical space. Now I'd like to introduce two of our fabulous speakers, two of some of the most brilliant, fun guys I know out there. First speaker we have is Dr. Vic Dutta. He has spent his career at the intersection of science, public health, and industry, driven by a simple belief: innovation only matters when it reaches the people who need it. As the global head of scientific affairs and industry access, food safety and quality at bioMérieux, Vic guides global strategy that connects breakthrough diagnostic technologies with the organizations and markets that rely on them. His work sits at the intersection of science, policy, and industry adoption, where effective leadership can accelerate meaningful, measurable impact. Our other speaker is Dr. Jose Emilio Esteban. Emilio is an accomplished leader in food safety and public health, bringing decades of scientific, regulatory, and international expertise in his current role as the Chief Scientific Officer at Marion Nutrasciences. He has previously served as the USDA Undersecretary of Food Safety, and yes, I know some of us are still working on forgiving him for that, and has provided key scientific leadership positions at both USDA and CDC, shaping national food safety policy and laboratory programs. Dr. Esteban has also been a prominent global voice in food safety, including his long tenure as chair of the Codex Committee on Food Hygiene and Service on the Executive Board of the International Association of Food Protection. Dr. Esteban's work in Codex has always, I think, stands as a stark reminder to all of us on how patient he is if he's willing to deal with Codex work. So let's give them both a very warm welcome, and we are certainly excited to hear both of your all's insights. Awesome. Well, again, thank you so much for both of you for joining us. We're excited to be able to set the stage with you and really have a conversation on why change is really needed. We're at a really interesting point when it comes to where we are at food safety. Are there different ways we can be looking at things? And I think we all are really motivated to not just testing or implementing programs that lead us nowhere. We're really driving towards our goal of improving public health. So I want to kind of start you all off with this discussion with this first question. The animal protein industry is facing increasing complexity from evolving pathogens to regulatory scrutiny and consumer expectation. From your perspective, what has fundamentally changed in food safety over the last decade, and why are traditional approaches no longer enough today?
May I go first?
Please.
Okay.
Absolutely.
So, Kay Rose, in one word, everything has changed. Okay, you said it yourself. I mean, the population has changed, the pathogens has changed, technology has changed, regulations have changed, and so what I really want to convey to you is we were very successful to get to where we are. Industry has been tremendously good at minimizing that hazard that's in food. And so what has worked before doesn't necessarily work for us to go to the next stage, right? We've gotten this far with what we did, but we need to have a new approach, a new thought, a new way of doing things to move on to the next stage of benefit.
No, fully agree with you. I would also say that the advent of technology and our knowledge, the amount of information we all hear about, things like AI, is making it more pertinent for us to look at these processes, even though we have done a tremendous good job. When I say we, I'm talking about the larger community as a public health community and also industry. We've done a tremendous job. I think it's a new lens that we have available now. So it's obvious for us to go back and say, all right, let's take a look at things that we may have missed in the past and or get ready for the challenges that are coming our way. So overall, I think we are sitting much better now than we have in the past, and it can only get better. So that's the good news, right?
Yeah, I think you both are so right. Another way um, of saying it that I sometimes refer to with our members, and it's like, why are we having to do some of these things different? Why, we've done so much already, we've done so well, and we have. I think the industry, like you both have said, has accomplished a tremendous amount. We've improved food safety and public health so much, and now our problems are a lot harder. We fixed all of the easy things, and so it's going to require more complex solutions, which I know we're going to get into today, to solve the issues that we're having now. And I think that is certainly well said by both of you. Much of the industry really relies on reacting once a presumptive positive occurs. Why is this reactive, hazard-based mindset that we traditionally followed for many years no longer sufficient? Again, it's that traditional thought of, oh, this always worked. Why is it necessarily working anymore? What does a truly preventative, risk-based approach actually look like in practice?
Right. Let me take this one first,
Sure.
if that's okay. So to me, when I hear hazard, I hear a static hazard, right? We're looking at something that is static. And we know that based on what Dr. Esteban just said, the world is dynamic. The food safety world is no different. The challenges are evolving, geopolitics, microbiology, environment. Our food is evolving. Our consumer base is evolving. And then the regulations are trying to keep up with it as well. So to me, I think in a dynamic world, to measure a problem through a static lens is right there. There's a disconnect. And so at the very least, I think the basic premise of hopefully what our conversation leads today is we're looking at a dynamic world with the dynamic challenges. At least we should be looking at dynamic tools that can also keep up with these challenges, whether it comes to testing, it comes to prevention, it comes to sampling, and so on and so forth. But the idea being like, let's not use a static tool to measure a moving risk. Let's use a moving targets to solve moving problems.
So I'm going to try to answer that by using an analogy. I think that's the right word. A reaction is when you get a picture and you see the picture and you react to that picture. What I want us to move is from that one single frame to a movie where we have frame after frame after frame, and it's the movie that we are actually reacting to, and then you can actually be more proactive because you know what is happening, what is leading to us to, and then you can actually prevent or direct what the next stage is going to be. And while we are doing that in the way of how we interpret the world, the way the world interprets us is also changing all the time. Influencers, the way we communicate, the way we receive information today, completely different than 10 years ago. And so we cannot use the same tools, the same approach, and always be able to react because that's never going to work in the future.
I think that's well said. Another way I'm looking at this is we're collecting so much data in everything we're doing. And I don't think data, I think very traditionally and in a very rigid mindset, people think of that as positives and negatives of some microbiological test I've done. But that's not necessarily that data is so much broader on that. And so let's deviate a little bit here. And can both of you talk a little bit about how important sort of data management and data in developing a proactive approach, how you really capture that sort of movie rather than a still picture? Especially, I think we often hear too, well, that's fine if you're one of the big guys, they have a lot of it. I don't have that as much. And certainly those challenges look different, but can you talk about it from different levels and sizes of establishment? What does that data picture look like, and how can that be catalyzed for better food safety?
Okay, well, I want to start with this one because as a regulator, I was receiving a lot of data all the time, right? But the one question that I always ask myself is, why did you collect this data?
Mhm.
So the message, the takeaway thing that I wanted to leave the audience with is, if you don't know what you're going to use it for, don't collect it. Just because it's available, don't collect it, because it could make things worse rather than better. And to the second part of your question about the different size establishments, in this country, the vast majority of the data points are going to be small or very small establishments. But the production itself is concentrated in the large ones, right? So granted, not everybody has the same level of resources, but there are creative ways in which they can be also get the same benefits. Where there is industry like this, sharing the good practices with the rest of them, but there's also, and I had just a meeting last week in Washington, and there's opportunities for grant mechanisms through other parts of USDA where they could be reached and helped to get to the same point. So I don't think that the size of the establishment should limit the quality of the food. I think everybody deserves safe food, and we should all be able to achieve it. But as far as data is concerned, you can have infinite numbers of data points, but unless you're going to use them, don't bother.
And I would go into the utility of the data itself, right? Small establishment, bigger establishments. I think data is, to your point, Katie Rose, is data. And it's one of those things that feed on itself. And we can, utilize it in a meaningful way to get better insights, right? And so again, I'm going to lean into the technology part where there are now ways to analyze this information, to then be able to see into the future, to see a dashboard them, and to be able to see what are the larger trends and make meaningful decisions, right, on a day-to-day level, but also into the mid to long-term level. And by the way, it's not just limited to microbiology. It is to truly understand the impact of food safety testing towards a larger operational efficiency and also the decisions we are making on a day-to-day level. All of that it's all tied through the underpinnings of data. And the more we dig into it, the more we find out. And so to me, that's the way to go. It's just a matter of how we get there. And this is where we often work with, your members or our customers. And then we see that the level of data that they're collecting matters. The quality of data that they have, what we call as data hygiene, is very different right now, right? Between smaller and bigger, it doesn't matter, right? It just comes down to, is that data usable? And so I would also say that, food safety testing, we always say, like, you cannot test your way out of food safety, which is true. But then in this process of food safety testing, we are generating a ton of data, which, to your point about, if stacked with the other metadata, it's giving us insights that we have never seen before. And I know we'll get into some of the other topics around the utility of it, but I'll give you one small example. In a lot of our examples working with our customers, we are realizing, or I would say the broader community, we are realizing that the amount of sampling that they're doing, it doesn't need to be done to that extent, because all of a sudden the data is telling us that your risk is moving, coming back to our previous point. And therefore, the amount of sampling that you're doing, and therefore the downstream testing that is needed to be done, also needs to evolve. So just improvement of the overall process control can be easily accomplished with the amount of data that already exists. And with slight tweaks and dashboarding and just building a larger insights around it, we can do a much better job.
Okay, Rose, if I can add just one thing before you wrap this question.
Yeah.
Now, when you are in a reactive mode, you're always trying to catch up. And so what I meant by the movie analogy was, we need to be part of the system, because the targets will not be the same, the pathogens will continue to change, the risks change. What's the point of sampling a surface after you cleaned it? Well, you cleaned it, right? So yes, you verify that it worked, but really, do you really need to repeat that? So I think we need to be smarter rather than more samples. Even though I work for a lab company, I would like to get all the samples that I can. You need to be smarter about the way you sample.
Yeah. No, I think that is so well said by both of you, and certainly appreciate. Related to that of what you're kind of collecting and the different things, how does focusing on biological insight and not just mere pathogen presence change that decision making?
Yeah. So I can jump into this one. the amount of insights we have gathered as a public health community in the last decade or so is absolutely immense. And I would split that into two different buckets. One is the pathogen itself. We hear about, agency or USDA and FDA doing, X number of genomes and all that. So just right there, we are getting information about these microbes like never before. And there is a conversation that is to be had around how can we utilize that data better. And so to me, the better one of the good ways of digging into that information is to look for indicators for the pathogens. And what that means is we are learning about the biology, the functional systems about these pathogens, and say, can we find a way to maybe detect these organisms in a meaningful way, right? So improving the indicators for a better indication, right? That's one way of looking at it. But on the other side, we are also realizing that, to Dr. Esteban's point, that the environment is moving, the microbiology is moving, and the microbes are moving. And so this is a ever-evolving process. So understanding a broader context of organisms and how they are reacting to different environments, different mitigations, and so on and so forth, is something is also we are learning more and more. The tools are getting better. We are able to understand a wider microbial ecosystem and therefore be able to also provide a better indication through better indicators. And in this case, the indicators are less guided towards pathogens. They can be, but I would say the overall quality and the hygiene of the processes and systems. Are they working? Are they not working? It can be now determined. And they all come down to the similar set of tools and data management algorithms and so on and so forth. The good news is it's all possible now.
So as you said in the introduction, I've been doing this for maybe too many years. But when we had the information that was lateral flow devices, presence or absence, that's what we reacted to. And then we got to PCR, and then we quantified. And now in 2014, we started doing sequencing. And every time we got a little bit more information, and that's great. I remember back in 2016, 10 years from about this date, approximately this date, we got together in WHO, FAO, and decided to classify Shiga toxin producing E. coli by genes and different levels of pathogenicity based on that. 10 years ago, well, guess what? The regulation is still considered them all exactly the same, right? But should they be considered the same? Back when we started with 0157, it was the one pathogen. If you look at the data from CDC, the vast majority of STEC was 0157H7. Well, today, if you look at the data, it's less than 30% of the STECs that are 0157H7. The vast majority are the other STEC groups, right? The other O groups. And so we need to find a way to look at the whole pathogenic group rather than just a specific serogroup. We have the ability to do it now. And if you want to be proactive, we should have maybe more refined approach of how to look at this. Same with Salmonella, same with Listeria. This is just the trend. If you get the genome, understand it.
Yeah. And there's a tangible example to that Dr. Esteban's former agency did end up adopting, which is, what we would call as a risk, like we as a, again, community risk-based test, which is where, to his point, Shiga toxin and intimin genes are part of the definition for the EHEX for the USDA, and also it's true for other governments across the globe. And finding those organisms that contain both have been a challenge, and that has caused unconfirmed positive rates, which is problematic to the industry and even any lab that is working on it, right? So because of the information, all these genomic information that has been collected, we now have certain gene targets that can predict the co-location of the Shiga and intimin on the same organism, which by itself reduces the unconfirmed positive rate, I would say at least 50%, if not more. And these tools are available in the marketplace. And so it's not a pipe dream anymore. These things exist, and I'm beginning to see similar things popping up in the world of Salmonella, and so on and so forth. So my point is that we are beginning to utilize this information, genomic and the genetic information that exists. So, and it's all very tangible now.
I'm glad that we're diving into that sort of more specific genetic testing and looking at different genetic markers and virulence, pathogenicity, and how we sort of evolved out of some of this traditional microbiology that we all know and love. Um, and I'd like us to dive, or like you two to dive in a little bit on that, on how that can help make more meaningful and sometimes actionable intelligence than sometimes very simple pass fail, and the type of things. And I'd really like to, in particular, have you guys address the benefits that an establishment who really adopts and embraces those approaches gain from sort of thinking about that differently and evolving their program.
Yeah.
So let me start on this one. Just imagine this. You're in a processing plant and you have so many combo bins and you test them each individually for whatever presence or absence of the pathogen. And you make a decision on that presumptive positive because you cannot hold the product until it confirms. Well, what if you only had to, and because of that product loss, when it's potential positive or presumptive positive, you divert that to a different, or you either hold it or divert it to a different type of final product with a reduced economic benefit, or eventually probably waste. If you can actually put another filter there that only takes care of that when you actually know that it's going to cause human illness, you will be saving a significant amount of product that would not go to waste, that would benefit the industry and would still benefit public health. So to me, having it rather than a hammer, maybe using a smaller chisel to look at things might be very, very useful with as long as we can do that decision at the same time, right? The one thing we can't do is wait another 24 hours, another 48 hours, another week. In the case of genome sequencing, another 10 days to get the sequence available, right? We need to be able to have that intermediate result that makes us be better risk managers that maximize the productivity of the company, but also maximizes the benefit for public health. So we need refined tools for that.
Yeah, food safety and operational costs are no longer mutually exclusive. They can be collective and actually sometimes compounds in both ways, in the short-term operational cost, but also you're gaining the efficiency in the operations. And there's multiple examples of that, and we're just getting started as far as, again, larger community is concerned.
One thing, Katie Rose, most of the things that we've discussed are not exclusively for the large producers. So for those of you that are online that are also smaller producers, this is not a differentiator by establishment size. This is something that's available to everybody at a reasonable rate. And so don't avoid it just because it's too complex or because it's too new or because I can't afford it. Let's talk about it.
Absolutely. And I would, again, another well said comments by both of you. I want to take again a brief deviation as we're in this space because we get this question a lot, especially from people who are first dipping their toe into this space. And really, there's a real big lack of understanding as how can you confirm that these pathogenic genes or these genetic markers that we're looking at and these targets are actually from the same cell? So a lot of speculation. Oh, if they're coming from different things, well, maybe there isn't a pathogen or an adulterant or whatever in there. How does the methodology and how does the test work that we actually can confirm that those are from the same cell?
Do you want to start that one?
Yeah, let me jump on that. So as test makers, I can tell you we take a personal responsibility to make sure that what we are rolling out and the claims that we are putting onto a test are perfectly vetted, right? So I'm just telling you our examples. I have no doubt in my mind that other test makers are doing a similar job. But typically, when we tend to make these type of statements that no, it's coming from the same organisms that contains both the virulence genes, we are talking about hours and hours of bioinformatic analyses to scrounge for every single genome that we can find on Earth. And I mean, not just United States databases, but we're also looking at, as far as Japan, to make sure that what we are saying holds true, because never say never in biology, right? So anything that looks too good to be true, we have to really, that much more data has to be put on top to make sure that it's doing what it's supposed to do. And then, so that's one piece of it. The second piece is the bioinformatics is a computational exercise. It has to then hold itself in the real world. And so there is as many E. coli's and other organisms that we're talking about, we can get our hands on, we do. And this is also true for not just the internal work. We would find additional partners who have expertise. These are world-renowned folks that we all, know well for their knowledge and expertise. And we will consult them, and we will ask them, like, what do you think? And how can you put it through the rigors to make sure it's done well? And by the way, all this gets then published, so none of this is like internal data. It's all peer-reviewed, available, right? And then the last piece is, rubber meets the road. So doing it in the actual samples that I know that our customers will eventually test, in the conditions that they will test, right? In the environment that they will test, with the technician skills that they would test. All of that comes together in what I would call it the robustness trials, to make sure that the genetics is holding, the test is holding, everything is working as it's supposed to. And then we obviously go to third-party validation agencies as well, and we say, you take a look at it, and if it works, give us the certification. So my point in telling you all of this is that the amount of rigor that goes into something like this is absolutely immense. And I can tell you, being involved in a few of these now in my life, that if there's anything humanly possible we could do to make sure that the test is doing whatever we are saying it's supposed to do, we do it. And if there's even after the launch, if some customer or regulatory agencies or, even yourself, if, come and say, you guys missed something like that, we have no problem backing into that data to make sure that it does what it's supposed to do.
Yeah, very little to add, but, in my current role, we actually use these kits, right? And we don't just use one brand or two brands. We use whatever is fit for the purpose that we're asking to test. Ultimately, we want to have the highest precision possible, but also we need to manage the turnaround times that are relevant for the industry, right? So for us, it's not just a matter of what we're testing for, it's why we're testing. At least in my role, I want to know. One of the things that I found out when I got into this new job is we give the customers what they want. And I'm like, well, shouldn't we give the customers what they need? But that requires having the conversation, right? Because what you want and what you need are not necessarily the same things. So for me, it's important to understand that whatever product you're going to run through your labs to service a client need actually is fit for purpose, right? And I cannot use that unless I myself in my labs, my technicians understand how that method works, its benefits, its flaws, its limitations, and its precision. All those performance characteristics. I would not want Mario to be using a system that we do not trust, because it's my name as much as anything else, right? Ultimately, we want to achieve a good test result fast enough, a reasonable price, but with the right answer, depending on what you're collecting.
Yeah, I would add, I want to expand on that word trust that Emilia just said. In a lot of good things that the industry has done, trust is one of those things. Trust between the industry and the consumers. We are consumers ourselves. We have kids at home, right? We ourselves want to be healthy. And so we would like to think that we are one of the stakeholders and continue to maintain that trust. And so going back to your point, Katie Rose, and you and I have talked about, the what would it take for us to do a good job when it comes to the new science that coming to, like, how do we make sure that all of us in the community are trusting of each other? That is a very important word, and we take that seriously. And like I said, it's not just us. I know this for a fact that other players in the testing market and the test manufacturer market, they all do an equally good job. And that's how we know that we are providing the right tool to the markets.
Thank you both. I totally appreciate that. And I know that's a long and complicated answer. And method development is one of those things where, again, we're answering complex problems. And so solutions and how we come to them, excuse me, can be very complex. So appreciate you both. Let's shift gears a little bit and talk about sampling strategy.
Mm-hmm.
Sampling strategy, frankly, I think is one of the most important things when you're talking about testing. What to sample, where to sample, when, how. From your experience, where do companies most often fall short, and what should they rethink to generate more reliable outcomes?
Okay, I'm gonna take a stab at that one first. Um, as we said before, things change continuously. Just because your sampling strategy worked last year, it doesn't mean that it works this year. Just because you sample everything and everything is negative doesn't mean it's always going to remain negative. And when you look at what you're sampling, it cannot just be final product. It has to include process control. Because if you look at statistics, the sampling power of testing final product is really not very high, because when you sample, you have to sample so much to make it relevant. So it's a combination of process control and final product. And then comes in the big question, right? How often? Where do I sample? How deep? What do I look for in my sample? And so I would encourage us to really have that conversation. And once you define as a company what your sampling strategy is, because you're the expert in your plant, because you know your business, because you know your flow, because you know your product, try to bring somebody else from outside to take a look, just to have that unbiased perspective. Because if you don't, you know that what you're doing is right because, well, you did it. You're the expert. You know your plant, right? Well, that's correct. And maybe 90% of the time that will be the case. But it's always good to have that other point of view, just to make sure that things have not been missed. And one thing I need to say is sampling more is not always better. I said it before, be smart about what you sample, how you sample, how frequently you sample. I can't, if you want to have 100% safe food, you sample all the food and then there's no food left to eat, right? I've said that before, I'll say it again. You cannot just base everything on final product sampling. For me, the emphasis, because it's proactive rather than reactive, is your process.
So it sounds like you think sorry, Vic,
No, go for it. Yeah.
it sounds, if I'm hearing correctly, you think probably the area where we're falling the most short of would be not necessarily being as intentional as we are with where we're sampling.
Right.
Yes.
Yeah, I was going to build with a practical example there, Kay Rose, based on our customer feedback and having spoken to our customers about concerns around sampling and the confidence that they have in their sampling. We end up building a service we call as adaptive sampling, and the name says it right there. Because of everything we just talked about in this webinar and something we have seen in the real world, which is the risk is dynamic, the adaptive sampling allows our customers to be smart with where they're sampling and how much they're sampling. Because the variables that impact sampling and thereby the downstream risks change from plant to plant, from season to season, commodity to commodity, and it requires additional look. And we have found savings for our customers without compromising the risk. Actually, we improve their risk profile because they're doing adaptive sampling. So again, this ties into a lot of things we were talking about, and I think I mentioned it in the data piece. It all starts with data. Our customers are willing to talk to us about what they're doing and having a conversation. They know their plants better than anyone, but then bringing us into the equation and saying, what do you think? And then we end up finding savings for them, again, without compromising the risk.
So, hey, Rose, you mentioned before what you sample. And remember, years ago, we were sending chunks of meat to the lab to test for 40157. And then we went to the N60 protocol that caused a lot of my employees to get, USA employees, to get hurt cutting, little pieces. And now we're going into this cloth sampling. And every time we change that, we can use the same detection technology. But if we don't know how we're collecting the sample and what we're collecting, whatever result we're going to get at the end from the detection is biased, right? It depends on what you did before. So you cannot test meat right after you've sanitized. You cannot test the surface of your sanitized because it's going to be negative. So the time, the what, the when, and the frequency all have to change depending on your unique situation in your plant, and most importantly, your goal. You need to have a goal before you start designing these things.
take-home point that we can never stress enough of the goal and to know why exactly you are sampling. So I certainly appreciate both of your comments on this. One thing that we talk about anytime we talk about sampling and testing, um, we have to talk about speed, right? As people more and more want to use the test to maybe make disposition decisions off of or do something with that result. Again, because we only test when we know what we're going to do with the answer. Can you talk about how speed being such a major factor for businesses' risk assessments and their operational decisions, how is the science evolving to support the industry in getting faster to results? And what impacts does speed have on that test result, whether it's especially when there's a presumptive positive that occurs?
Yeah, I can speak to the science part, and I'll let you speak to the operational. The science is evolving incredibly fast. If not already, there is quite a few systems or companies and startups that are providing solutions for faster time to result. And just to be clear, what that means is it is a combination of sampling tools themselves. You heard about cloth as one of them. And then the chemistries that are involved in making sure that we are able to detect or get enough DNA from the organisms upon either short protocol or sometimes without it, that is then allowing for a faster time to result. And so at this point, the test being the hold for time is getting that time lag is getting smaller, if not eliminating, depending on what are you trying to do with the test itself. So we are getting to a place now. Where can we go with this information? If I have a test in 6 hours or maybe right away, what does that mean? How do I make my decision differently? And I'll let Dr. Esteban jump into that.
Yeah, for me, we were discussing this with one potential customer, and they said, well, I need the results during my shift. And I asked, well, do you want your result during the shift, or do you need your result during the shift? Because if you're gonna ship every 16 hours, if your trucks are lined up for shipping out every 16 hours, you need your result 15 hours, right? You don't need it 8 hours or 6 hours or 4 hours. So yes, people normally want, and it's the nature of human beings. They want it faster, they want it cheaper, they want it better. Wouldn't it be lovely? Yes, it would be, right? But when do you really need that according to your business process? So move away from the pathogen, move away from the precision. When do you need to make that business decision? And that should drive what type of methodology you need to have. Granted, the sooner you know your result, the more flexible your decisions are. So I'm not going to argue that part. But really, do you really need that immediate result? If you're not going to ship that product for the next 8 hours, are you willing to pay more for that immediate result if you're not going to do anything with it for the next 8 hours? So we're living in a world where you can have a huge menu of analytical solutions. And so you need to pick from that menu the one that fits your business process. I hate to take science away and look at operations, but ultimately you need to use the data point, not just generate it, right? So to me, it's a combination of things. But yes, faster will be more convenient because it gives you more options. As long as that faster doesn't mean less precision, because then it's bad information.
Right. I mean, Katie Rose, do you hear a lot of need from the industry for the faster time to result?
Oh, absolutely. Everybody always wants it. Again, like you said, faster, more accurate, and cheaper. That is, and I think especially with certain pathogens and where some of the regulatory landscape is going, this pressure to have a faster result turnaround is becoming increasingly important to establishments. And I think, how you talked about it, Emilio, of this vast menu of things you can pick from, it is so true. And it can be so overwhelming. It's overwhelming for anybody, whether you're a big guy who has a big staff of a lot of brilliant scientists, or if you're a small guy who you're one guy wearing three different hats, your food safety, your worker safety, and you're maybe the comms person, you might be all. It can be very overwhelming. And I think one thing I'm hearing from all of you is there's the options are out there, right? And knowing that how it works is there's always going to be trade-offs. If you want something faster, it may not be 100% accurate. That doesn't really exist, but you know what I mean. You might give up some on that sort of accuracy and precision. So what level meets you and your business needs for what type of test you're looking at? And I think that's where we always try to drive home to everybody. Your laboratory person or the lab you're working with should be your partner in helping make these informed decisions of what works for your business. It is their job to know all these different menus of tools and say, okay, let me help. Or that's where you find a really good laboratory partner to come in and say, let us help you figure out what is best for your business to make those decisions with. Awesome. Well, I know we are getting close on time, so I want us to finish with one question before we open it up to the audience, which we already have some questions coming in. But so I want you to leave the guys group. If there's one sort of mindset shift you'd like industry leaders to take away from our discussion today, what would it be when it comes to microbiological testing, not as a checkbox, but maybe as a strategic tool for continuous improvement?
Yeah, I can say that more testing is probably not the right answer. And therefore, data becomes that much more important. So if I were to leave our leaders today with an idea, I would say, please take a look at your data hygiene, the extent of it, because that's a gold mine that can help you save a lot of headache. But also economically, it can be really, really helpful. And then how to go about it? You just said it, Katie Rose. We are here to help, and they can always reach out to us, and we are more than happy to sit down and have a very pertinent conversation to their context.
So for me, the main shift I would hope to generate from this conversation is clarity of purpose. Why are you doing the work that you're doing? And then pair that with transparent and open communication, right? If you actually trust what you're doing and you're willing to share that, everybody, the community is much better. In your position, I mean, you have all these producers that are with you, and you're much more powerful if you all work together than if you work in individual establishments, right? The global solution is one that comes from multiple sources, not a single one. So the mind shift is share the information, share your experiences, but have clarity of purpose when you apply any type of lab methodology.
Absolutely. Thank you. Well said. I think that's why one of the best things that the industry did is many years ago made food safety a non-competitive issue. We
Yes.
are here. Rising tides raise all boats. We all want to sell and provide safe products to consumers. We're all in this together. And so with that, we are going to open up to our larger Q&A. Please feel free if you've got a question down at the bottom of your screen, you will find the Q&A feature. Please go ahead and plug any questions you have in there. We have had some that come through. So as other people are typing those in, we're going to go ahead and start with those questions that have come through. Let's talk about how we address moving targets. Question came in. That makes it ever so hard. And I think there's sort of this moving targets as far as I want to answer this in two ways. I want to answer it as moving targets of the food safety risk within your establishment, and frankly, moving targets as it comes to regulatory pressure. How does industry address moving targets with side of both of those lenses? I think the answer might be different depending with both of those thoughts. So I'll let you two decide who goes first.
Well, I'm the regulator. I'll go first.
Please.
No.
Recovering regulator.
You said it, Katie Rose.
Former regulator. When I have a moving target, I need to focus on what I intend my food sampling program to do, my food safety program to do, and that's to offer safe food. Okay, so I need to focus on the things that are going to cause illness. So for me, it seemed natural a decade ago, and it seems more natural today, that I should look at the things that are actually causing me to get sick. And when my technology limited that to she got toxin present in some type of food, that was one thing. But now I can actually tell which cell has which genes on it. And so that solution when I address the pathogenicity of whatever pathogen it may be, Salmonella, Listeria, E. coli, whatever it is, Campylobacter, whatever, if I can address a single solution that focuses on the broadest of targets that is more comprehensive of my challenges, that's what I would do. That's reactive. You still are looking for the pathogen. On the proactive side, I would then go and say, okay, just I have this information for the pathogens, I have the same information of the microbiome that favors those pathogens to be part of my environment. Can I look at that microbiome outside of the sample that allow for that Salmonella, E. coli, or Listeria to be present? So find a lowest common denominator that gives me more coverage. I think that would be the simplest solution for something, for a moving target. You throw a bigger blanket.
Yep.
Awesome. That's helpful. And I want to like dovetail off that a little bit too, because I think it's related to moving targets. But moving targets is kind of what you were talking about adaptive sampling. That's kind of you trying to chase down this moving target a little bit with your adaptive sampling of trying to figure that out. I think there was a question that came in is how do we explain or have that conversation with our inspector or the regulatory officials in their establishments on when they're looking at the data of again, are you chasing a problem? Do you know of a problem that you're having with this adaptive sampling? And so give some tips on how can someone engage in that conversation with their implant personnel.
Wow. Our inspectors do a phenomenal job, in my opinion. Well, our inspectors, the FSIS inspectors, I'm still looking at that environment. Anyway, an FSIS inspector does a phenomenal job with the tools that he or she is given and an environment in which they have to operate. So I think when you try, the best thing for the plant management to do is to be able to build a relationship with that inspector and actually, I'm not say educate, but have the conversation as to what you're doing, why you're doing it, how you're doing it. Because the adaptive sampling concept, if you really look at it, statistical and things like this, it's going to be difficult to convey. You have 2,000 establishments, 2,000 inspectors or more. You're going to be impossible for you to say the exact same message to all 2,000 of them, right? So the consistency of the message is going to be different. But the one thing you can all do is be able to have those conversations to try to do a common level of education and conversation that the rules are interpreted at least closely in two different establishments, right? Ultimately, is the human factor that comes into play. The inspectors are going to always cite a CFR or a section of the CFR as a flaw. And that's inflexible because it's the way it is. But the interpretation of that is human. So build relationships.
And what also helps, Cairo, this is real world experience, is that collation of the information, and I'm talking about different layers of metadata, and then dashboarding it, making it known that in the world, in the sea of things that we are doing as an intervention, such and such, to your point about beyond the relationship, this is the problem we're trying to solve. It's not that we are chasing, it's like an emergency everywhere. It's just like, look what we are trying to do. And then contextualizing it with, it's not a problem we're trying to solve. It's more like we're getting efficient. And this is why all those tools now are available for the establishment to be able to use on a regular basis. And they are very communicative. They're not meant to be, you don't need a data scientist on the staff to help explain some of these trends and analyses. And again, going back to your earlier point, this is where the lab partners, as well as the test manufacturers like ourselves, can come in and help, translate some of this information. And then it comes down to, like you feel better about your data. I mean, it's already like you already have plenty of information, but the fact that you just take all this complex information and you create a simple dashboard from it just makes a sea of difference.
Okay, Rose, let me just very briefly here.
Mm-hmm.
If you're going to deal with an inspector or with any other human being, for most of us, a picture tells you a lot more than a lot of text, right? So if I have a map of my plant that highlights heat map or whatever you want to call it, distribution, where my sampling was, why it was negative, why it was positive, that's such a unique tool to be able to have that conversation with the inspector, right? It's not this table and this page and this report and PDF file. if you actually have a way to show them in a picture what you're trying to do, that requires you to have that conversation, and then they feel part of it, right? So to me, it's using the tools that you have available today, with dashboarding, with visualization tools that we didn't have many years ago, right?
Yeah, absolutely. I say this all the time. I think if you can color code something, it all of a sudden just becomes so much more digestible, including my calendar. If I can color code my calendar, I all of a sudden miss less meetings. So I hear you. Next one, Vic, I want you to start with this one, partially because I can punish Vic now that we're no longer co-chairs of NACMIF and I don't have to talk to him 3 times in one week. I can be a little meaner to him and put him on the spot.
Okay.
This is kind of a good broad question, and I think very cutting edge. How do you see AI being used and helping us incorporate into our testing programs? As far as where, when, how, what's the future of that look like, you think?
Yeah, it's all evolving, and the evolution is not just on the AI technology itself, which I'm sure y'all are aware of how much investments are going into it. And I heard someone say it's like today is the dumbest AI ever going to be. So let's just start with that as like the bottom line, right? So we can expect it to get better into the future. But then the second piece I keep coming back to, AI doesn't run by itself. These algorithms work from background data. And then the old adage, crap in, crap out is very true now, and probably more so now than ever before. That said, the world of test makers are already utilizing AI to make so those algorithms that we are deploying to make that call between yes and no, it's a form of an AI, right? It is not an AGI. It doesn't think on itself, and that's where I think the gap is, and we need to be going. I can foresee the idea of adaptive sampling, getting overlaid or getting powered by AGI down the road, where the human intervention becomes less needed, which means we can then go do other things, and there's plenty to do all the time. But these are to me, I think in order to get started, it requires a good quality data, good quality metadata, right? So this is where I go back to my take-home message. Please take a look at your data. And then let's talk, because the tools are evolving, the information is moving fast, and the future is bright, but we have to work together, like you said very well.
One thing, if I can expand on that, I believe in AI. I think the world is going to be more and more using this, but never forget HI, human intelligence, right? You still have to look at that information. I don't care how refined your AI is, you still need to use the human intelligence to be better. And in that sense, I forgot to mention in one of the responses, but we also need to include academia in some of these things, right?
Yeah.
We it's not the regulators, industry, and consumers. Academia plays a significant role, and our extension services have been really, really hammered lately. And so I don't know how to include it in one of the responses, but I had to mention that our academic partners are also a key collaborator that we should not forget.
Here, here. I certainly think that's well said. Now, in the context of rethinking food safety, um, do you think any of the focus should shift maybe from more extensive testing to sanitation and food safety culture? How important are those components in an overall food safety program?
I think they're essential.
Yeah.
Without a sanitation program, an effective, validated, and verified sanitation program, the food safety system is going to be very, very challenged. So, and I wouldn't say they're separate; they're the same thing. For me, the sanitation program is part of a food safety system, right? And the targets are different, and the interventions are different, and the reaction is different. But you have to consider everything under the same umbrella. Sanitation is key, but it's only part of the whole approach.
Awesome. I have 2 more, so please be quick on this next one so we can get to the last one as well. And this one, I think, again, I'm going to put you back on the spot, Vic. Can you expand some thoughts on microbiome analysis tools for the future? 16S RNA sequencing. Can these be valuable assays without regulators using the data against the industry. I think there's always some question. People hear the word sequencing and they think whole genome and it's going straight into NCBI. What can 16S do for us to answer those some of those questions out there? And how do we not have it be created as a bat to use against us?
Yeah, the 16S, the resolution on it is typically not deep enough to make a confirmation on pathogens. Now, there's a caveat there, so I will not go there. But also, when you're looking at the microbiome population versus a colony, which is what WGS is, you've already identified or you have confirmation on it that it's salmonella. Now you simply just, beating the heck out of it on sequencer to say what type of salmonella, right? Microbiome and 16S sequencing is much broader, and it's much, I would say, it doesn't go as deep. And the purpose of microbiome 16S based sequencing is to get an understanding of a microbial population at a genus level, typically not going down to the species and then further down, right? How does it impact the overall? We are finding all sorts of use cases. You may know, there's companies out there that are doing this commercially as a service, and I also know a lot of the folks are already using this information already to assess their spoilage, assess the overall shelf life, and so on and so forth. And I strongly believe the science is not quite there yet, that there are indicators embedded in this information that will point us to the risk factors, not necessarily the presence or absence of Listeria salmonella, but it'll point us in a direction that would be meaningful to then intervene through better sanitation procedures and whatnot, and all that information. The good news is it's no longer an academic exercise. It is something that your members can reach out today, and it can be a reality tomorrow. So that's where we are.
Awesome.
I'm glad you were brief.
I'm sorry.
Yeah, I know, right? Final question. You both have, 15 seconds each.
Okay.
Oh, I got it.
We have talked about bringing in outside opinions to kind of help in different partners in food safety. How do you recommend people finding the right outsider to come in and help evaluate and improve their process?
I mean, don't go with the first opinion. You have the ability, you have the resources, you have the power to decide who you're going to be your partner. Just remember that your partner is partner for a longer time, not just for that one event. So invest in the short term, but with a long-term perspective. Find the right team, you'll be much more successful.
I will also be shameless in this plug to say, also reach out to here at the Meat Institute. We have our entire team in Reg Services who does this a lot of times. So please reach out to Norm, Roy, or Clarissa, or Casey Gallimore and myself. We know people. It is our job to know people and connect you with who you need to. When in doubt, please reach out to here at the Meat Institute. Well, again, thank you, Dr. Detta and Dr. Esteban, for your fantastic conversation today. We sincerely really appreciate you and also really appreciate the support of bioMérieux, both for the industry and the partnership you do with many, many of our members to help improve food safety, but also your partnership with us here at the Meat Institute. The Meat Institute has more great educational content coming up. Please check out our events page at the Meat Institute website. The QR code in the slide will take you directly to that events page, so I invite you to scan this on your phone to check that website and see what's coming up next so you can sign up. This concludes our webinar today. We hope to see many of you again in the future, and thank you again to bioMérieux and all of you for joining us.